Hello everyone, and welcome to today's GCSE Biology revision session. In this video, we'll be exploring microscopic units, a key part of understanding magnification and measurement in biology. This is the first topic in our Microscopes and Magnification series, so let's get started. By the end of this video, you should be able to recall the SI units, which is French for International System of Units, used in biology for magnification, and convert between SI units used for magnification. In everyday life, we measure things using units like meters, centimeters, and millimeters. These are ideal for objects we can see and touch, like the height of a door or the length of a pencil. But in biology, we often explore structures that are far too small to be seen with the naked eye, such as cells, organelles, and molecules. To study these, we use microscopic units. Before diving into the microscopic scale, let's quickly review some familiar metric conversions. There are 1,000 meters in a kilometer, 100 centimeters in a meter, and 10 millimeters in 1 centimeter. Most of us will be comfortable converting between these units, but when we get into the smaller units, we can get confused. A tip to remember is the acronym MIS-DIL. You can use this to guide your conversions. Multiply when converting into a smaller unit, and divide when converting into a larger unit. This simple rule helps you move confidently between units. To help you visualize these units, here are some biological examples. A nerve cell can be up to 100 centimeters. The pupil of your eye, the dark bit that takes in light, typically measures between 2 and 8 millimeters. A red blood cell is around 6 to 8 micrometers in diameter, and the diameter of a DNA molecule is approximately 2 nanometers. Finally, a carbon atom is incredibly small at just 70 picometres across. These examples show how each unit is used to describe structures at different scales, from whole cells down to individual atoms. Now let's go through how to convert between these units. This is a skill you'll need for your exams, and it's easier than it looks. A key rule is to convert to a smaller unit, multiply, and to convert to a larger unit, divide. Think of it like money. Imagine you have £10 in the form of a £10 note. If you break it down into £1 coins, you now have 10 coins. So, if you're going from notes to coins, you're multiplying. Multiply if going to a smaller unit, miss. If you're going from coins to notes, you're dividing. Divide if going to a larger unit, DIL. Now apply that to the microscopic units. 1 millimetre is equal to 1,000 micrometres. 1 micrometre is equal to 1,000 nanometres, and 1 nanometre is equal to 1,000 picometres. We can use a visual representation to simplify and guide our unit conversions. So if you're converting from millimetres to micrometres, you multiply by 1,000. Just like turning one £10 note into 10 £1 coins, miss. If you're converting from micrometers to millimeters, you divide by 1,000, just like exchanging coins back into a note, DIL. If you want to convert between other units, simply replace the units. Place the larger unit on the left and the smaller unit on the right. This can be done for any unit conversion. You may want to take note of this before we attempt the practice questions. Let's try a few examples. A student observes several plant cell specimens on a microscopic slide. Complete the following calculations. There are 3 questions, and each question has 1 mark given for the answer and 1 mark given for the unit. Question A says, cell X is 0.11 millimeters in length. Convert this into micrometers. Step 1 is to identify the direction of your conversion. We are converting to micrometers. a smaller unit. So we need to multiply by 1000. Step 2 is to actually complete the calculation and ensure that we include the correct unit in our answer. So 0.11 millimeters times 1000 will give us an answer of 110 micrometers. Always check that your final value is labelled with the appropriate unit. This is essential for exam marks. Let's try the next question. B. Cell Y is 76 micrometers in length. Convert this into millimeters. Again, recall step 1 is to identify the direction of your conversion. We are converting to millimeters, a bigger unit, so we'll need to divide by 1000. Step 2 is then to complete your calculation. Originally, we had 76 micrometers, and to convert that into millimeters, we will divide by 1000, which will give us an answer of 0.076 millimeters, including the units. Last practice question now. C says, cell Z is 92 micrometers in length. Convert this into centimeters. Similarly with step 1, we always identify the direction of our unit conversion. We will initially convert to millimeters, a bigger unit, so we will need to divide by 1000. So 92 divided by 1000 will give us 0.092 millimeters. Step 2 this time is we will then need to convert the millimeters into centimeters, a bigger unit. So this time we'll need to divide by 10 because there are 10 millimeters in 1 centimeter. So 0.092 millimeters divided by 10 now gives us 0.0092 centimeters. Step 3 is to ensure that not only have we completed this calculation, but we have put in the correct unit for our final answer. Now that we've reviewed how to convert between microscopic units, it's time for you to have a go yourself. A microscope image shows a cell that is 0.0035 millimeters long. Convert this length into A. Micrometers, B. Nanometers, and C. Meters. Your task is to remember to follow the steps that we practiced. Identify the direction of conversion, then multiply or divide as needed using your visual aid or the acronym MISS-DIL. Include the correct unit in your final answer. When you're ready, pause the video and take a moment to work through each part carefully. Check your units as you go and press play when you're ready to see the answers. Right, let's go through the answers. The answer to A is 3.5 micrometers. The answer to B is 3,500 nanometers. And the answer to C in 2 steps is initially to convert the millimeters into centimeters, the answer being 0.00035 centimeters. And then finally to convert those centimeters into meters. Because there are 100 centimeters in a meter, we divide our answer by 100 this time, and that gives us a final answer of 0.0000035 meters. I hope that you got those correct. If there's anything that you want to be reviewed again, feel free to rewind the video. Right, well done. Today you've successfully learned how to record the SI units used in biology for magnification and convert between SI units used for magnification. Very soon you'll be able to access the revision page and exam style questions linked below to reinforce what you've learned. If you found this helpful, please like and subscribe to stay updated with the rest of the Microscopes and Magnification series. See you next time.